Alright, this online video is on the scientific method. Here's your list of objectives. Remember, as you go through the online video, make sure you keep these objectives in mind and that you revisit them at the end to make sure that you understand everything that was covered in the lecture. So, in order to start talking, we need to address science. Since science in general is derived from Latin meaning to know. So it's a pursuit of knowledge, and it's how we ask and then find answers to questions. And this is questions about the natural world around us. So therefore, we're limited to what kinds of questions we can ask, because we can only design experiments for questions that can be tested and that we are able to design an experiment for. So for example, religion is something science will never claim to try and explain. So an example of a question that science would not ask, science would never ask, does God exist? That's not something that can be scientifically determined, and therefore it is out of the realm of our studying. Also, when you go through the scientific method and you think about something scientifically, it helps to reduce an emotional reaction, because emotions are a form of an opinion, and science is all about the seeking of truth or knowledge. So it has nothing to do with what you hope the outcome will be, but rather what the numbers tell you from an experiment or what the data shows you. So that's going to be really important. Sometimes there are scientists that can let their judgment and their desires for a certain outcome cloud their ability to do science properly, and that's when we get into problems. For example, there was a scientist several years back from now really wanted to be known for making the first human clone, and he kind of lied about it in his paper, and he made the data look as though he did it successfully. But what happened is there was an investigation, and they found out that he actually fudged his data. So he made some of it up because he just wanted really badly to be the first scientist who was able to clone a human. He was removed from the scientific community. He was fined a lot of money, and he was actually thrown in jail for fraud. So it's really important that whatever the data shows us, we stick to it. So in science, we talk about a method, and I'm sure in middle school you went through it. And I know that the notes go step 1, step 2, step 3, step 4, step 5, but understand that the scientific method is a process that is it's cyclical in a way. It can go in and out of the observation phase. You can loop through experiment, analysis, and then go back into another hypothesis. So don't think of it as very linear, but rather think of it as a cycle. So we're going to go through each part. Okay, step 1, making an observation. When you observe something, it is using your senses, and a lot of times kids focus on using their sense of sight, but don't limit it to that. For example, I'm recording this early this morning, and I can tell that even though I have blocked my windows, the sun must be up because I hear quite a lot of activity going on from the birds. Or I can smell that in the other room breakfast is being cooked because it's an odor that I am familiar with. So it's a way of using your senses, and it leads to posing a question, or we call it also identifying a problem. So to go through this PowerPoint, we're going to use an example. And the example we're going to say is, I wonder if flea collars really work. You might be familiar with flea collars. You put them on cats or dogs, and they are supposed to give off a chemical that repels fleas. So people spend a lot of money to make sure that their cats or dogs have them. There are companies that only make flea collars, and they make a lot of money off of it. So it will be really interesting to see, do they really work? Okay, well, all we did was ask a question, do they work? But the next thing is to form a hypothesis, and that is what we predict the outcome will be. So it is our explanation for an observation. And there are some rules as to how you do a hypothesis. It must be written in an if-then format. And we'll kind of come back to this when we talk about the experimental design, because there are two key parts to it. It definitely must be something we can test. For example, something that is out of the range of testing within our classroom is to determine if an alien spaceship is the reason why some dogs don't have fleas, because like the spaceship emits like an exhaust, and that kills all the fleas in an area. That's not something we can test, okay? And most of the time it's based on prior knowledge. So I'm sure some of you have heard of flea collars before. Have any of you seen them on other pets? That might lead you to say, oh, well, they must work. But a scientist never assumes. We always go through, we test, we collect data, and we analyze from our experiment. So a properly formatted hypothesis in this example: if I give a dog a flea collar, then it will have less fleas than a dog without a flea collar. So we're identifying what we're testing and how we're going to test it against something that doesn't have what we're testing. And we'll get into the terminology for that in the next slide. So now we're ready to design an experiment. We have an idea of what we want to do. And we need to identify our variables. So these are things that can change in an experiment. And you always want to make sure that you have one independent variable. Independent is the thing that the scientist is trying to test, or we're purposefully changing. We also call that the manipulated variable. Your teachers may have used that term in middle school. In high school and in college now, it is standard to use the term independent variable. So in our case, the independent variable would be using the flea collar. That is what we're purposely changing about the dog. Okay? The dependent variable is the part of the experiment that changes in response to the independent variable. So you may have heard it called a responding variable. So in other words, by using a flea collar, what measurement is going to be different compared to a dog that doesn't have a flea collar? And we would look at whether that dog has fleas or not. So being able to identify the IV and the DV for any experiment is going to be really important. So our IV in this experiment is the use of a flea collar, and our DV is going to be the presence of fleas. But not everything about an experiment can change, or how do you know if the independent variable is the thing that caused all these changes? So we need to have controls. So your control group is going to be your group of test subjects that do not receive the independent variable. And a lot of times students get that mixed up with a controlled variable, or a constant as they can be called. And these are the parts of the experiment that stay the same. So your control group is going to be a group of dogs that do not have the flea collar. That will be your control group. Controlled variables are things that need to be the same about both groups of dogs, because as you can see, we're sort of formulating an experiment where we have this group of dog gets the flea collar, this group of dogs don't get the flea collar. Okay, so we know that that's the one difference between them. So what should be the same between them? Well, sometimes your controlled variable list can go on and on and on. You always want to think about the fact that, in an experiment like this, you want to keep some environmental factors the same. So you want these dogs to all be boarded in the same way. Maybe they're kept in a kennel with the same cleanliness. You want them to have the same food and water. You want them to be bathed the same number of times per week. You want them to have the same amount of time playing outside. You want them to, basically be the same sort of grouping, maybe dogs all of the same age, or that they're all mutts or a variety of breeds. Lots of examples of controlled variables. So things that you want to stay the same. That way, when you get a difference in the data, you're not sitting there going, well, was it because of the flea collar, or was it because sometimes these dogs were let outside more often than these dogs? You don't want to have to choose between that. You want to know that the only thing that was different was that independent variable. So here's an experiment. I said, let's take 500 dogs, because you want to have a good sample size, a whole bunch of different breeds, and they're all from local shelters. So these are not domesticated dogs. They're not, living in houses. They're living in, a kennel. The vet is going to choose 200 dogs that have a high number of fleas to begin with, and is going to randomly assign them to two groups. You don't want to have any purpose in that because unconsciously you might start putting certain dogs in certain groups. It's really hard to do things randomly. That's a challenge for scientists. So we're going to make sure the dogs are boarded in the identical environment and that they're treated the same, the only difference being that IV. So one group of dogs wears the collar and the other group of dogs does not. And it's really important that this design can be reproduced because every time someone else does this experiment, you want them to come out with the same data because that helps to make yours more valid. And then you can say with more confidence either yes, flea collars work, or no, we can't confirm that flea collars are truly better than not having a flea collar. So let's say we've done the experiment, now it's time to analyze data. Well, what kind of data did we collect? You have a choice, okay? And there are different types of sciences and different types of data. Qualitative data, you should see the word quality in there. Those are data points that cannot be measured numerically, so they're not numbers. So they're a little bit more variable in how a person can judge them. So for example, let's say you and I were sitting next to, a dog, and we were measuring the amount of itching behavior. Okay, I might say dog A showed less itching than dog B, but you might have interpreted some of it as just as much, and you might say that they both itched about the same. There's a lot of variation in how we determine behavior, and because of that, it's something that we can't assign a number. Quantitative data, seeing the root word quantity in there, okay, those are things we can measure with numbers. So for example, we can physically count the number of fleas that each dog has. And we can say, okay, there were 4 fleas on the dogs that had the collars compared to 500 fleas on the dogs that were in the control group, meaning those that did not have the flea collar. In general, for our type of science, the hard sciences, biology, chemistry, physics, quantitative data is going to be the best. It's the most reliable. If I have a dog and I say, okay, I counted 26 fleas, you should also count 26 fleas. But if I point to a room and I say, the color of that wall looks blue, you might say, well, I think the color of that wall kind of looks more purple than blue. So there's a lot of variety in qualitative data. Quantitative data is much more reliable. Everyone measures it the same way. So after you have that data, you can model it using charts or graphs, something to display and look at trends or patterns. The last part here is coming to your conclusion. So you want to go back to your hypothesis and either accept or reject it. Okay? We don't say that we have proven a hypothesis. Proof in science is a pretty dangerous word. It means unchanging. We know for certain. And while we like to say we have a lot of certainty in science, we never know. Okay? This was one experiment that we did. Is there a small chance we did something wrong? There could be. So we're going to accept the hypothesis and we're going to use our data to support whether we accept or reject that hypothesis. Another part of the conclusion that's really important is identifying sources of error. In other words, you're trying to tell your audience, listen, I did everything I could to make sure that this experiment was performed perfectly, that all of my controlled variables remained constant, that everything went smoothly, and I analyzed the data without putting my emotions into it. However, if something went wrong, and if I came to the wrong conclusion, here may be some ways that I could have changed the data unknown to me. Okay, and the most common is human error. All right, if let's say your job is to measure out 25 milliliters of a substance, okay, and you pour it into a graduated cylinder. Well, I'm eyeballing it, and it looks like 25 milliliters to me. But maybe it was actually 25.2 milliliters. And depending on the experiment, that error in measurement could have a huge impact on the outcome. Problems with equipment and also having additional variables that we don't know about, that often complicates experiments. So in this dog flea collar example, let's say, oops, we didn't look into the history of the dogs that were in the local shelter, and it just so happened that in Group A, a majority of those dogs had only been at the shelter for the past 2 weeks, that they had been given away from homes. And so they would naturally have a lot less fleas than a dog who's been there for months. And if that's something we didn't look into, that could alter the way we interpret our data. So why did we do this experiment in the first place? Because your teacher said so, right? But scientists who do this as a living, and earn money for it, they have to get money in order to do their experiment. So they have to show that these results can be important to society. So, cancer research gets a lot of funding because there are a lot of people who think it's an important thing to research and that we need to find cures and ways to deal with it. But a lot of people don't find that it's important to do research in undersea caves to determine if this very rare sea slug is on the brink of extinction. So we need to find an importance for society or a way to link it to your everyday life. So what do you do now that you're done? You made all this conclusion, you stood up, and you said, look at how important my results are. Well, sometimes the results can raise more questions, and that's going to lead to more experimentation. So there's where you see that cycle happening. It's also important to repeat the experiment. Again, that helps to verify your results and really solidify and increase your confidence in accepting or rejecting your hypothesis. And you want to spread the word. You want to share your results. There are lots of ways to publish them in scientific periodicals or magazines. And also, scientists like to go to lots of conferences and share their work with each other. So you might be familiar with the peer review process that you use in writing class. Professional scientists, grown men and women, also use the peer editing process before they hand in a publication. So the community relies on one another. And that's a really important part of the scientific method. So that's the end of this online lecture. If you have any questions, feel free to email your teacher or come see us in class.